I think you're muted. Oh.
I may start again. So, good afternoon, ladies and gentlemen, dear colleagues. A warm welcome to our HealthTech Lecture, Dynamic Matrices, a material platform for precisely controlled cell culture and ultra-selective biomolecule isolation. It's my great pleasure to introduce our speaker, Dr. Elisha Krieg from the Leibniz Institute of Polymer Research in Dresden. Dr. Krieg is a chemist by training. He studied chemistry at the University of Cologne and the Weizmann Institute of Science in Rehovot in Israel, where he also received his PhD in chemistry in 2013. He then continued his scientific career as a postdoctoral fellow at Harvard University in Boston, followed by an open topic postdoc position at the Technical University of Dresden and the Leibniz Institute of Polymer Research, Dresden. Since 2020, Dr. Krieg has been leading the working group DNA Nanotechnology, building programmable materials and nanometer scale devices at IPF Dresden. This group develops materials with programmable and reconfigurable properties, using DNA not only as a carrier of genetic information, but also as a highly versatile construction material. By combining DNA nanotechnology with polymer chemistry and molecular biology, his work aims to transfer nanoscale programmability into macroscopic material systems for biomedical applications. Today's lecture will be about two systems. One is DyNAtrix, a fully synthetic matrix for 3D cell culture that enables precise and dynamic control of the mechanical environment surrounding living cells. And the second is LASSO, a new approach for the ultra-selective isolation of biomolecules designed to achieve high purity and efficiency while reducing complexity and costs. So we are very eager to learn about those systems. Dr. Krieg, thank you very much for joining us, and the floor is now yours.
Yes, thank you very much, Jens Hellwager, for this nice introduction and also for the invitation to give the talk here. I'm happy to tell you a little bit about dynamic matrices, which is an innovative material platform for cell culture and ultra-selective biomolecule isolation, two applications that don't seem to have anything to do with each other, but which are connected by a common technology platform. Second. Yes. Yes, I'm a group leader here at the Leibniz Institute and also a young investigator of Technical University of Dresden. We are located in this very nice building, the Max Bergmann Center for Biomaterials in Dresden. But as a disclaimer, we also received recently funding from Exist Forschungstransfer, which hopefully will allow us to commercialize some of these technologies that I'm showing you here. So this prospective startup is called Dynamic Matrices. It's under constructions. I'm a co-founder, so just for full transparency and as a disclaimer, and you will also notice during the talk, while this talk is mostly focused on the academic research side, once in a while I will reveal my interest in this company and into the commercialization efforts as well. So, of course, as a scientist, I'm interested in academic research about publishing papers and creating patents, all of which can be summarized as kind of output. But what is even more rewarding, I think, to see also some kind of outcome in the form, for example, of a company that enables more people not only to read our papers and to cite us, but also hopefully to use this technology and to benefit from it in the future. So as chemists, we're very excited and proud of our synthetic material world. And I think, of course, in healthcare, this is true as well. Synthetic materials are incredibly important, whether we use them inside the body as implants or on the body or for in vitro studies. They really have revolutionized healthcare. But when we compare what we have in the synthetic world with biological materials that make up our body, we have to recognize that these two material types have highly different properties. Biological materials are information rich. And with that, I mean they contain information in the form of their high compositional complexity. We have tens of thousands of different proteins in each of our cells, essentially. Our cells encode via their genome very different behaviors. They can differentiate differently and adapt to different stimuli. In the synthetic world, we have information-poor materials. We have materials that are compositionally minimalistic, 1, 2, or maybe 3 different components. To keep it simple and to prevent these components from interacting with each other in unforeseeable ways. And these materials are also fairly static. So the challenge is oftentimes that how do we make these two material types to interact? How can we make synthetic materials that interact predictably and in a smarter way with biological systems? Can we make synthetic or information-rich synthetic materials that programmably interact with biological molecules and living tissues? That's the question that really drives my research and the research of my group. To address this challenge, we rely on a very special molecule, which is DNA. And of course, all of you know DNA as the general carrier of information, all living organisms. We are interested in synthetic DNA as a building block for materials. And that's because DNA has this very unique property that it sequences information also very faithfully encodes its binding interactions. You know this from biology classes in high school, that G always binds to C, A always binds to T. And the beautiful thing is that not much else is going on in terms of intermolecular interactions. And that's the reason that methods like DNA origami or DNA nanotechnology have been so successful and really revolutionized nanotechnology. So today we can design nanosystems that contain hundreds of different DNA strands, which we pipette together all, and they all self-assemble and to find their position and to create something like this nanoscopic smiley face. And this really has revolutionized nanotechnology. But in my group, we don't want to make more nanostructures or smiley faces, but we want to use the exact same principles of programmable self-assembly to make soft materials with DNA-encoded physical properties. So I will show you these two technologies that benefit from this new engineering paradigm. One of them is DyNAtrix, a programmable and fully synthetic 3D cell culture matrix. And the second one is LASSO, highly selective biomolecule capture and release. I will first start to talk about DyNAtrix. And if you don't care about cell culture matrices, cell culture, or material design, you can skip this part and come back in half an hour when I talk about LASSO. So 3D cell culture is really a transformative technology for biology and medicine. We can take cells outside of the human body and study them under very well-controlled conditions in a petri dish. We can grow them even into organ-like miniature versions that are called organoids. And this, of course, is very powerful for basic research, for example, to address questions in developmental biology. We can carry out drug screening. We can model diseases in a petri dish. And also, more and more, there is the hope that we can use this technology to develop personalized therapies. For all of this to work, we need a very special material, which is a cell culture matrix. The most widely used class of cell culture matrices are basement membrane matrices. You may know them under brand names like Matrices. These are liquids. Originally, liquids come in these little flasks, and they're actually derived on a massive scale from mouse tumors. So it's one of the largest uses of lab animals is for growing enough tumor materials that provide enough of this basement membrane matrices for cell culture. When you then take this liquid, mix it together with cells, you create a gel. And of course, now this gel is supposed to mimic the kind of environment that the cells experience inside the organism, to pretend to the cells that they are still inside the organism so that the questions that we ask the cells are actually relevant. And of course, as you can imagine, this comes with a lot of problems. First of all, since we are dealing with animal-derived matrices, they are usually not very reproducible. That's something that users always complain about. This comes also with regulatory limitations that prevent the progress into translational medicine. And of course, these materials also entirely fail to mimic human tissue mechanics, because the human body or the extracellular environment doesn't behave like this gummy bear here. It behaves partially elastic, it behaves partially viscous, and behaves partially plastic. And there is currently no cell culture material out there that can faithfully reproduce and kind of to independently tune those different properties. If we look at biological tissues, we see that they actually experience or exhibit highly different mechanical properties. They vary vastly on the spectrum of storage moduli. So storage modulus is G prime, or what people typically call, quote unquote, the stiffness. They vastly vary in their loss modulus, or G double prime. And tissues are usually not perfectly viscous, nor are they perfectly elastic, but they're somewhere in between. And for a long time, when researchers were referring to the mechanical properties of their cell culture matrix, they usually would just refer to the stiffness, but kind of ignore other mechanical properties, which now are starting to emerge to be much more and recognized as much more relevant, and maybe as relevant as the stiffness itself. One of these parameters is the stress relaxation behavior. So that's the time-dependent behavior of living tissues, where, let's say we take this gummy bear again, we put a certain strain in the laboratory on this gummy bear, and now we can measure how much does the gummy bear actually push back against the instrument. In the end, that force will be large, and then it will decay over time. And that's what you see in the stress relaxation curves here of actual tissues. You have brain tissues that have vastly very short stress relaxation time, sub-second stress relaxation. And then you have tissues like skin or muscle that have much longer stress relaxation times. And what is important to recognize is that this is really a parameter that is independent of the stiffness value. So you can have materials that are have slow stress relaxation, like this gummy bear, or fast stress relaxation, like this Play-Doh piece here, but still identical stiffnesses. And just like we as humans, if we interrogate these materials in our hands, for example, we notice differences between these materials in terms of stress relaxations. The cells will notice differences and will behave differently in these different materials. So to address the challenge of tuning these types of different mechanical properties and bring more functionality into cell culture matrices, we developed a material platform that is called DyNAtrix, and which we believe represents the first programmable cell culture matrix. DyNAtrix contains two major components: a synthetic polymer on the one hand, and DNA modules on the other hand. And the way I like to describe these two components is through this analogy. The synthetic polymer here acts similar to this screwdriver here. It defines a certain range of applications, but then specific functionalities and properties are introduced by the DNA modules, and they're exchangeable and provide a very rich spectrum of functionalities. Our synthetic polymer is a ultra-high molecular weight acrylamide-based polymer. We have DNA strands covalently linked to this polymer, and their only purpose is to bind other DNA-based modules. And then we have other biofunctional signals, most importantly, like adhesion peptides. We use a lot of the RGD adhesion peptides that provide anchorage sites for cells to mechanically interact with a matrix. Now, all we have to do in the simplest case is adding a crosslinker. So we add a crosslinker just by pipetting, no chemistry involved, no photoactivation involved, and that crosslinker spontaneously assembles with the anchor strands and then strands bind to each other to form what we call an overlap domain. This is a very large crosslinker if you compare it to other crosslinkers that exist in typical hydrogels. But the presence of this overlap domain here that has a specific DNA sequence, usually between, let's say a dozen of nucleotides long, it encodes a large number of different parameters. For example, thermodynamic properties, the kinetic properties, things like the melting temperature, also mechanical stability, and the network topology. And this is a richness in design space that no other crosslinker can provide you. This project was started by a very talented master student, Yusuan Peng. And very early in this development, we found that it was highly useful to not use a single crosslinker, but diverse libraries of crosslinkers. So complex mixtures of libraries of crosslinkers that we add to this polymer. They assemble with a polymer at one temperature and then further assemble. And through the diversity of the crosslinkers, we can actually suppress intramolecular crosslinks and thereby enhance the presence of intermolecular crosslinks, which makes the crosslinking much more efficient and reduces the amount of DNA we actually need. And that's important both for cost considerations and also for compatibility considerations. One really cool thing about using complex crosslinker libraries is that we can now use the library complexity to change the elasticity of the network. So that's a unique mechanism. Usually, hydrogel elasticities are controlled by crosslinker concentrations. We can also do that. But in addition, we can control the elasticity or fine-tune the elasticity just by the complexity of the library, by the number of components inside the library. And this is a property that can be very nicely also simulated. So there are large opportunities for just predicting the properties of the material prior to an experiment. Most importantly, we can also control the stress relaxation behavior in a highly systematic manner. And that's because the mechanical rupture force of DNA duplexes is highly predictable. So if we take a duplex and we pull it on the two ends, there is a certain nanomechanical force where this duplex will just open up, reversibly snap open. And if we bring them back together again, they will close again. So this has been very well characterized already in single molecule experiments. And we use this very unique property to generate what we call stress relaxation crosslinkers. So this is just a set of different crosslinkers that can be plugged into the gel on demand. And when we plug these different crosslinkers into the gel, we obtain gels with vastly different stress relaxation times just by a couple of nucleobases that are here in this mechanically weakest link. And we can control stress relaxation times from anywhere between sub-second times to stress relaxations on the timescale of hours or even days, which covers the entire range of stress relaxation times found in all living tissues. Now I want to tell a small anecdote about what happened when we first started introducing cells. Of course, when you start introducing cells, there is the problem that you can't just anneal the material, but you have to strictly stay within a physiological range. In this case, we needed to stay below 40 degrees. And so we can't just anneal the material. So the problem now arises that DNA hybridization is extremely fast. And our thought was that, well, all we have to do, if we go back here, is to mix together this ultra-high molecular weight polymer. We mix together DNA modules and cells together really fast, and then the gel will just form around the cells. The problem is that this binding happens extremely quickly. So crosslinkers almost immediately form. And no matter how fast the student was pipetting together these different components, you would always end up with a non-inhomogeneous kind of mess. As you can see here, this is a two-component system where one component was fluorescently labeled, and you can see it's absolutely not well mixed. And it would make a very big difference for a cell to sit either here in this gel or over here in this gel. So using this programmability of the crosslinker, we now saw that it's important to control the kinetics of crosslinkers. So we introduced this little blocking strands that binds to the overlap domain initially, but they're designed to be labile enough to fall off at a specific threshold temperature. So these were designed to, block the initial hybridization at 4 degrees, so we can mix everything together. And then once we heat the system to 37 degrees, the proper temperature for cell culture, the blockers spontaneously fall off, and now the cross-linking can happen. And this really led to a much more homogeneous and mechanically well-behaved hydrogel. And very nice side effect is that now we have a workflow that perfectly mimics the workflow that people are used to from cell cultures in things like Matrices, where also you warm up the solution and now you have your gelling, now you gel your material. So DyNAtrix has high viability, or cells embedded in DyNAtrix have high viability in these tests, higher than for 2D cultures, and even outperforming Matrigel in most of these cases. What is really nice is that we can expose the cells or the hydrogel at any point in time to nuclease. So for example, DNase bonds that can be added. It diffuses into the gel, and then it degrades selectively the cross-linkers. And this is a very mild mechanism by which cells can then be released back and kind of analyzed downstream. Well, if the nuclease degrade DyNAtrix, one may ask, and we actually get asked a lot, this question, is that DyNAtrix not just degraded or digested by nucleases that are commonly found in the culture medium? And to answer this question, here is a DyNAtrix gel. When we expose it to nucleases as that which are found in this, for example, in serum supplemented, FBS supplemented media, and if you do this culture, then indeed you see a fairly rapid degradation of DyNAtrix within 24 hours. Of course, at one point, this degradation stops as the nucleases are kind of exhausted. But this initially indeed was a problem. But fortunately, there are various agents that we can use to inactivate nucleases. For example, actin is one of those nuclease inhibitors. It selectively binds DNase 1. And we can actually use actin to either drastically slow down or hibernate nuclease degradation or adjust it to a level that we want. For example, if you want the matrix to be slowly degraded, you can kind of tune that rate of degradation by the addition of actin. So in actin supplemented cell culture, as you can see here on the right side, the cell culture was stable. Here, this is a video for 24 hours, and typically current protection strategies that we use stabilize the matrix for up to 7 days in presence of serum. If you don't have serum, you can grow these cultures for up to 60 days. So there is actually no degradation happening from nucleases that would be secreted by the cells. Over the years, in this initial study, we validated already a good broad range of cells and organoids with which DyNAtrix is compatible with. So this was stringently tested with MSCs, iPSCs, kidney cysts, and placenta organoids. We also have various other collaborations that are really important for us to continue expanding the compatibility of the matrix. So we have worked with brain organoids, intestinal, kidney organoids, for example, and also at the moment we are really interested in cancer spheroids. I want to highlight one study just to exemplify why DyNAtrix can be very useful for people working in cell culture, in particular people who are interested in effects of mechanobiology. This is a study we did in collaboration with Alf Honigmann in Dresden at the Biotech, where we looked into the polarization of this MDCK. So these are canine kidney cells, which form these cysts in the presence of RGD in DyNAtrix, we form this nice apical insists that are expected and which also would grow in matrigel. But if we have a DyNAtrix backbone that is lacking this RGD peptides, we actually get these kind of non-typical apical out cysts. So clearly, cell attachment and mechanical interactions are critical for the correct polarizations of the cells. So knowing this, PhD student Xuanku Zhao conducted very systematic studies in DyNAtrix where he varied independently the stiffness stiffness, G prime, and the stress relaxation time, tau. So we have 12 different combinations of stiffness and stress relaxation times. And what we see here is that the polarity or the ratio of apical in cysts is strongly a function of the stiffness. It goes down with decreasing stiffness, but it's also strongly a function of the stress relaxation time. So you can have a situation where you have a stiffer matrix with a shorter stress relaxation time that gives you similar results as a longer stress relaxation time with a moderate stiffness level. So this is already quite interesting. So what is actually happening here? In order to understand this, we have to understand what is actually happening in the cell. So cells are connected to the extracellular matrix by these transmembrane proteins called integrins. They bind to RGD sites in the matrix. And only under tension, these integrins will undergo conformational changes that then recruit additional adapter proteins, ultimately focal adhesion complexes, and ultimately connect the actin cytoskeleton to the extracellular matrix. And that is also what is needed for many cell decision-making processes. If the tension is sustained, for example, in slow stress-relaxing DyNAtrix, we get this downstream signaling. But if the tension is decaying too quickly, so fast stress relaxation time, the conformational changes in the integrin won't be maintained, and we will only have very weak downstream signaling. And so that's exactly what is happening here. Now, the big question is actually, what timescale is relevant for the cell, right? Does the cell decide on its polarity on the timescale of seconds or subseconds, so meaning fast timescales? Does it interrogate the environment very quickly, or does it take a lot of time, for example, hours or even longer times to assess the mechanical environment? This is a question we could then answer in DyNAtrix because we have morphological outcomes, which can be represented as a vector. And we also have the stress levels that are present or would be present in our matrix for a continuous range of timescales. This is given by the stress relaxation times. And now we can correlate those both parameters and see what timescale actually matters most to the cell. So we can scan through these different timescales. For each of these timescales, we obtain correlation coefficients. And when we plot this correlation coefficients over the continuum of time, we see that actually the 1 second or sub-second timescales do not matter so much for the cells. But what matters much more are timescales in the range of 1 to 3 minutes, so 100 seconds, or 3 to 9 hours. So this is beyond 10,000 seconds. And what's interesting about this is that when people compare their hydrogel matrices with each other, they usually refer to 1 Hertz values, which are kind of would represent this timescale of a second. But what we see from this data is that what is much more relevant, at least for polarization of cells, are much longer timescales. And what is really needed is a full characterization of the viscoelastic properties of the material. Of course, we had this conventional stress relaxation crosslinkers that allowed us to set the stress relaxation time at one point in time at the beginning of the cell culture, but we wanted more. We wanted to be able to also control the stress relaxation at any point in time during the cell culture. In order to achieve this, we upgraded our stress relaxation crosslinkers into a switchable version. The switchable stress relaxation crosslinker looks very much like the conventional one, except for it has this blue domain, a regulatory domain. And this can be actually addressed by DNA-based signaling strands. So if we integrate this switchable SRC into our hydrogel, the hydrogel will have a certain stress relaxation time, and now we can modulate that time with signaling strands. Signaling strands that shorten the stress relaxation time are called fragilizers. They add it to the gel, they diffuse into the gel, they bind to all of the crosslinkers, and now they have this additional green domain that can invade the overlap domain, which is, of course, weakening then the overlap domain mechanically without opening it entirely. So this comes into a different stress relaxation state. Now we can reverse this process by adding a strand that we call the reinforcer. The reinforcer binds the fragilizer and then removes it gently out of the crosslinker. It creates a waste product that then diffuses out of the hydrogel and restores the original state of the gel. And with this, we have a mechanism that allows us to, at any point in time, switch the stress relaxation time between two or more different values. We tested this with these MDCK cysts, where we showed that switchable stress relaxation can guide the polarization of these cysts. So we have an input here that is our stress relaxation time. For example, we can switch it from slow to fast and then leave it as fast, or switch it from fast to slow and then go back to fast. So as you can see, we looked at different permutations here, and we see that the cells very nicely dynamically respond to the modulation of their mechanical microenvironment. So we can switch between these two different states. That would be the output here. And it's really a unique way how we can control the development of cells without biochemical signals. We change biomechanical signals. These are videos of this process actually happening in real time, or not in real time, but in these cells. These values here are ours, and as you can see, these cysts remain in the apical state for quite a while, and then the transition into an apical out polarization is very fast. And in this paper, we also show that we can not only switch between the different apical in versus apical out, but we can also achieve certain states of the cell culture, for example, this non-spherical apical out or multi-lumen apical in, through switching back and forth between different states in a way that wouldn't be achieved by just statically leaving the gel at one particular in one particular state. Of course, we can use DyNAtrix in different formats. For example, we have now recently begun using it on this microfluidic chips, which make switching, for example, much more easy, and it's fully compatible and provides the exact same results. One nice property of DyNAtrix is that it is self-healing after mechanical breakage. That's what we show in these rheological experiments, where we kind of torture the gel through 10 breakage and recovery cycles. And as you always break just this mechanically weakest overlap domain, this breakage is fully reversible, and you can restore fully these mechanical properties of the gel that the gel had initially. This is important because if you want to print or inject a hydrogel in an extrusion-based manner, you need it to be self-healing. And what we show here is that you can just extrusion print or extrude DyNAtrix through a tip. At the end of the tip, the matrix will become liquid, but then it solidifies directly downstream as it exits the tip. This is important for extrusion printing, so we demonstrated that we can print 3-dimensional tissue scaffolds. And importantly, because the gel liquefies at a certain mechanical force, the force load or the strain on the cells is extremely reduced. So the cells are protected, and this leads to a very high viability of about 98%, in this case, MDCK cells again, that were printed on a surface and showed very high viability after the printing process. Now, sometimes I get used, can DyNAtrix be used in vivo? And we actually want DyNAtrix to replace in vivo for at least for things like drug delivery, but we have run some rudimentary immunocompatibility and blood compatibility experiments in our original study, which show that DyNAtrix is both non-immunogenic and hemocompatible when exposed to whole human blood. So we don't see significant immune response, and also we actually see a decreased coagulation activation of DyNAtrix, which is not due to the DNA, but actually due to the acrylamide backbone, which are actually superior to both glass and to Teflon. There was one recent study, and I have to stress here that this was totally independent of us. We just saw it from the literature, and they used DyNAtrix and kind of reproduced it independently for the treatment of post-stroke brains, for post-stroke brain regeneration. So these were experiments in rats where they induced a ischemic stroke. In this case, immune cells and inflammation and dead neurons are found in this stroked region, and they inject as a treatment DyNAtrix, which was loaded with neural stem cells and engineered mesenchymal stem cells, and they found that in this setting, DyNAtrix supports neural differentiation of NSCs and mitigates inflammation. So at least that is consistent with the results that we have with whole human blood, and seems to indicate that DyNAtrix can also be used or may be a promising material for in vivo experiments. All right, we have various functional modules now, combinatorial crosslinker that give us provide access to stiffness, heat-activated crosslinkers that allow control over kinetics, stress relaxation crosslinkers that give us vast control over stress relaxation times, and these switchable crosslinkers that let you, the user, change the properties of the hydrogen matrix anytime you like. One last module that I want to mention, and which is not yet published, and nevertheless we are very excited about this, are so-called force probes that can be plugged in now into the material and allow us to map mechanical forces in 3D space and time. And so this is a brand new module. These force probes are homogeneously dispersed inside the gel, and whenever one of these force probes breaks, it creates a fluorescent signal whose lifetime can be controlled. So it allows us to see mechanical activity of cells in 3D space and time. We tested this, and this was a collaboration with Dr. Anna Taubenberger and Dr. Alf Honigmann, both from biotech. Here we tested this in invasive breast cancer cells. The yellow signal is the actin cytoskeleton. The pink signal are actually our force probes. So we see how the force probes are activated by these punching cancer spheroids. Once in a while, cancer cells get ejected out of these spheroids and then migrate through the matrix, migration paths that can be tracked. We can track kind of force fields around cells that are spreading inside the matrix. And we can also kind of semi-quantitatively investigate how or where mechanical stresses or strains are building up around these spheroids. So we believe that this can be a very powerful method for studying the role of mechanical forces in development and disease. So hopefully this will come out in the next month or so on bioRxiv. Okay, I want to summarize the advantages and also make sure that I mention some of the current limitations. Advantages are we have really an exceptional and unmatched control over matrix mechanics anytime during cell culture. The material is fully synthetic, compositionally defined. xeno-free, so no animal-derived components in there, which makes it highly regulation-friendly if you wanted to do anything else than research-only use cases. The material is gently degradable, and we can apply it in different formats, in classical cell culture dishes, microfluidic chips, or 3D printed. We have demonstrated now continuous culture for up to 60 days. And we have validated various types of, or we and others, let's say, have validated this various other types of cells and a couple of organoids. Limitations are, if you're used to working with Matrigel and whatever you throw into Matrigel will form an organoid, that if you want to do that with DyNAtrix, that has to change. You need to know what your cells what your cells need, because DyNAtrix is just a blank slate framework in which the cells have a well-controlled mechanical environment. It doesn't have the biochemical richness that Matrigel has. So the trend would be, or the important transition would be, towards a system where all components are known. Secondly, long-term cell culture, while it's feasible, it's only feasible in nuclease-free media. So our nuclease inactivation strategy works for up to 7 days, but if you need substantially longer times, you should avoid the use of serum or conditioned media. Again, it's better to have a fully defined, fully chemical medium with chemical composition. And then also our stiffness, so the G prime is currently limited to about 400 Pascal. So we can't yet make materials that are really stiff, like kilopascals or tens of kilopascals, which might be used for certain cell types. With that, I want to make you aware of our DyNAtrix pilot user program. If you're interested, you can get a DyNAtrix kit and just test it whether it works for you. We have 2 different kits. The ready-to-use kit, that's just a fully synthetic matrix with pre-adjusted parameters that, let's say, should work for most cells, and at least viability shouldn't be an issue. If you're interested in the advanced mechanical tuning, you could try the minimal discovery kit, which gives you a range of different stiffnesses and different stress relaxation times, like similar to in the paper that I had described to you. How do you participate? If you want to participate, if you're interested, please shoot us an email to contact@dynamicmatrices.eu and tell us just a description of your application and problem, what outcome you would ideally hope to achieve. Then we will look into it. We will tell you, give you an honest opinion. Yes, this can work, or no. If yes, you will receive a pilot kit. You can use it with your own workflow. And then we hope that you can give us a roughly 10-minute feedback when you're done, to let us know how it went, whether it worked. We are always happy to have suggestions for improvements, more ideas, and general feedback. If you're interested in one of the more advanced features, so this is like fancy stuff like switchable cross-linkers or force probes. This we don't have yet pilot kits for. And if you're interested in that, please write to me, and we'll see whether we can collaborate or apply for a common grant. All right, this is the part about DyNAtrix. Now I will shift gears and talk about LASSO, highly selective biomolecule capture and release, which seems to be a very different technology, but you will have a couple of déjà vus, I think, that will remind you of DyNAtrix. So in healthcare, here I've just listed a couple of different applications we typically would encounter, clinical diagnostics, drug discovery, genomics, and transcriptomics, and all of these methods involve some type of biomolecule isolation, right? And these are, of course, extremely important, but what one should not forget that each of these also needs some kind of sample preparation. And even though the analysis arena has developed quite rapidly, if you think about the revolutions in genomics and transcriptomics, I wouldn't say the same thing about sample preparation. So sample preparation kind of relies on decade-old and very battle-tested, robust methods. For example, chromatography, which is like from the 1950s. on one hand, which is about half of the market, and then you have more new things, modern things like bead-based pull-down assays, particularly popular are these magnetic bead-based assays, which are from the 1990s. And this is actually a fairly large market. So alone the nucleic acid isolation and purification market is about currently $8 billion and is projected to grow to about 13 billion by 2030. And even though this is, of course, very important, the status quo is still really plagued by a couple of fundamental limitations. What are these limitations? First of all, materials that are used for biomolecule isolation usually have limited binding capacity. We usually use solid particle-based systems that can only bind their target molecules on their surface. So the bulk is basically just additional weight. Many commercial systems also have very fixed target selection, so we can bind DNA or RNA or proteins, but usually not all of them at the same time, and oftentimes also just a subset of these different molecules. And then finally, and maybe most crucially, there is a pervasive problem of off-target binding, which is inherent to solid-liquid interfaces. So every solid-liquid interface, like a microparticle, exhibits some non-specific adsorption. It's similar to your surfaces at home catching dust. It's inevitable. And even though there are, lots of anti-fouling agents or anti-adsorption strategies, this is a recurring problem. So a lot of time is wasted and money is wasted, and we believe that we can do much better than this. And I also would like to note that when we have kind of non-specific binding, we introduce biases into our data. And then now that more and more data is analyzed by AI in much larger quantities without human supervision also, all these biases and distortions in the data can lead to problems in the AI interpretation, which may pick this up as a trend or a signal. So that's also why we need more clean samples and clean data. So a former PhD student of mine, Sarah Speed, developed this really cool and very different biomolecule isolation assay that is called LASSO. LASSO is supposed to be very efficient, universal, and most importantly, ultra-selective. And the way it works is that instead of a solid particle, we actually have our DNA functionalized polymers. These are exactly the same polymers, except for the RGD peptides or biofunctional motifs there that we use in DyNAtrix. So it's really the same material platform. We have exactly the same cross-linkers. And then we have an additional module, which is catcher strands. So these catcher strands are added to this material to define the binding properties of the material. These polymers are used at a much lower concentration. So instead of forming a gel, the cross-linkers now program the phase separation properties of the polymer. So the polymers bind targets, then they phase separate. And as they phase separate, like oil droplets separating from water, we can spin this down and kind of remove all the contaminants from the supernatant and reobtain a pellet afterwards. This is the process actually happening as observed in a confocal microscope. It looks a little bit like it's snowing, but what you see there is not solid particles, but these are highly swollen kind of gel blobs, 99.8% water. The rest is polymer, and the fluorescent signal that you see is the fluorescently labeled target molecule that is selectively fractionated into the phase-separated polymer blobs. So no solid interfaces. Ideally, we shouldn't see any adsorption that is typical of solid interface in biomolecule separation. You can spin this now down. This is again fluorescent images. And importantly, if you leave out the catcher strands or you leave out the cross-linkers, neither the target nor the polymer will be deposited. So it's indeed highly selective. We can do this for DNA We can do this for RNA, both through complementary oligonucleotide catcher strands. But we can also do this for protein via aptamer strands. So aptamers are DNA or RNA strands that selectively have been selected to bind proteins. The first application, and kind of the killer application that we saw for this system, was the depletion of ribosomal RNA when we do transcription profiling, which is of course very important for both fundamental biological research and also diagnostic applications. We typically get total RNA, and then we put this sample into a sequencer, typically in an Illumina sequencer, which then gives us the transcription profile, just the abundance of each of these transcripts. But a typical transcriptome actually contains vast majority of ribosomal RNA, rRNA, which we are not usually interested in. So depletion of RNA is commonly done to remove the ribosomal RNA. And when you do that, you can actually drastically enhance the detection of all other transcripts. So this is a very important method. Almost always, or some version of this is used for kind of selecting the signal from the noise. This is a surprisingly large market, about $250 million just for RNA depletion or rRNA depletion kits. So we benchmarked LASSO against the two most widely used RNA depletion kits that also work very fundamentally differently. SI-Tools RiboPools, which is magnetic bead-based assay, and NEB's NEBnext enzyme-based assay. They are both in the order of about $50 per sample, which is actually quite expensive, and a lot of costs for the sequencing are going into kits like those. What I can say is that LASSO can be produced orders of magnitude cheaper and can be sold, we believe, for about $15 per sample, which is of course about three times lower than that. And that's possible because LASSO is relying on just a simple synthetic polymer rather than these recombinant enzyme containing kits, which also needs, of course, cooling and cold storage, all of things that LASSO in principle doesn't need. When we benchmark this against these two kits, we see that LASSO, first of all, as it should do, increases quite drastically by a factor of 4.3 the number of detected transcripts, which is very comparable to the RiboPools kit, a little bit lower than the NEBnext kit. However, the NEBnext kit is really sensitive to degradation, so you need to make sure that it doesn't expire. So the moment it's below its expiration date, beyond its expiration date, it strongly reduces its efficiency here. What's the biggest advantage of LASSO from our perspective is the selectivity, which indeed produces up to 7 times fewer expression outliers. And of course, these are data points that would create problems in data interpretation. Here, just as an example, here the expression profiles, what you see here in this data clouds are several 10,000s of transcripts that are compared to their original undepleted sample. And what you can see in all of these three methods is that they kind of increase the number of counts. So this is this shift upwards, thereby increase the detection of these low abundance transcripts. But what is really important is that in the speed-based assay, RiboPools, we see a lot of these orange data points. So these are non-selectively depleted transcripts that were not supposed to come down. And of course, this is a big problem. We see this also in NEBnext kits, maybe a little bit less than in the case of the RiboPools. However, we see a couple of severely depleted transcripts that actually are completely lost, and some of these are diagnostically relevant markers, for example, microRNA cancer markers. This advantage of LASSO over these two commercial assays is also reflected by this correlation coefficient, showing that this distribution of data points, which ideally should lie on the perfect line, is much closer to that perfect line than in the case of these two other kits. We can also, as I mentioned, pull down proteins and also release them. So we tested this with a human thrombin, which is bound to an aptamer. And our catcher strand also contains this regulatory domain. Again, it's a little bit similar to the regulatory domain in DyNAtrix for stress relaxation switching. Here, what we're switching is the release of thrombin. So we can add a release strand after thrombin pull down, which then kind of throws out thrombin from its binding interaction with the aptamer. And what we can see here from this plot is, first of all, we do not have any non-specific capture of thrombin. We have very high pull-down efficiency of about 90% in this case, and we have near quantitative recovery of captured thrombin from the matrix. And this thrombin is fully catalytically active, as we can see from the cleavage activity. So very gentle capture and release mechanism. So in summary, for us, LASSO outperforms the commercial pull-down methods. We have about 8 to 20 times higher binding capacity, so more more effectiveness of the material that is being used, 7 times higher specificity. The assay needs about 15 minutes hands-on time. We have at least 3 times lower cost, and it's a very gentle purification method that is applicable both to RNA, DNA, and also to proteins. Also here, in the case of LASSO, we have a pilot user program. So if LASSO is something that you are interested in, you can either approach us for getting what we call RiboLASSO. So that's a LASSO version that is optimized for rRNA depletion, kind of ready to use. Or we also have a LASSO Flex version that kind of only contains the polymer and cross-linkers, and you just get the information of how to dock your specific kind of target capturing module, and you have total freedom of designing your own pull-down assay with this. Again, if you would like to participate, shoot us an email, describe your problem and the application, what outcome you would like to achieve. We will evaluate this idea. You will get a pilot kit, use the kit in your own workflow, and then hopefully you can give us a 10-minute feedback on the usage and how it went. Okay, with this, I described to you these two technologies which kind of conveniently use the same material platform. So we can do certain synthesis workflows or combine certain synthesis workflows of backbones and components of these materials in our labs and kind of split them up. And this is quite efficient for us and gives us a lot of flexibility, and just exemplifies the advantage of this programmable material platform, similar to DNA origami. You can make a smiley face out of an origami, or a rectangle, or a triangle. And here you can really address very different applications with one and the same material platform. With that, I would like to thank first and foremost my wonderful group. So Krishna, who started as a master student, did PhD, and now is a postdoc, and he's really leading the business side of our spin-out plans, and who's very active also on our social media channel. Peng and Tim, who both pioneered and developed DyNAtrix, who are also, like Krishna and myself, co-founders of the company. Sarah Speed, who unfortunately left us back for the US, but still we are in touch, and she really pioneered LASSO. And Yannis, who is now a new team member and who has really already contributed a lot on the further development of LASSO. So we are of course supported by our great institutes, our funding. I have to say, big shout out to our collaborators, without whom we wouldn't be able to actually do any meaningful, cell culture work. And I should also say thanks to our industry supporters that have either provided, very useful discussions, support letters, that had basically in grant applications, or even support us for a specific project financially. With this, yes, and of course, very importantly, I would like to thank you, especially if you would like to become a pilot user, a collaborator, an investor. We'll be happy to talk to you. Thank you for your attention, and I'm happy to take any questions.
Yeah, thanks a lot, Elisha. That was very good to learn so much about the two technology platforms, and especially also that you have this pilot user program. I think this is so helpful to get into the communities, and of course also for you to probably learn a bit more about things that you haven't thought about or you haven't done in practice.
Absolutely.
So wonderful overview and very convincing stories. Thank you. So before we come to the questions from the audience, I would like to know, so you said that you started an exist program. Will this be more about the LASSO approach, or will this also combine the two platforms, or what are you exploring there?
So the exist was gained through DyNAtrix, because that was the first that we had the feeling this is market ready. Within the first, so the exist runs for 2 years, and within this year, the LASSO story also developed very nicely. And since it's the same material, we would like to also explore this option. And there is always the question, like how broad can we be in terms of our research, focus. And I think that's an advantage for me, being an academic group is, in principle, we can explore all kinds of things. Of course, the company has to prioritize, but I would like to explore both of these things. And for LASSO, we actually would like to acquire also more academic funding. Yeah. And the LASSO actually is supported by a small grant from Leibniz Health Technologies, I should mention, which is really important because it supports one position. Yeah.
Yeah, thank you. Thanks for mentioning that. Although this is a smaller contribution, but on the other hand, I mean, you showed already the situation with the competitors, and you made a clear offer that you can beat the price and even increase the quality, and showed a clear market. So in the end, as you said, it's a similar background. I mean, in terms of substances. There was a first question to the first platform, or we start with the first question. And the question there was about the DyNAtrix. Did you add some linkers to bind proteins to enhance the cell adhesion?
In the DyNAtrix backbone, we only have the kind of peptide-based side chains. We thought about, it's a good idea, we thought about adding additional binding sites for specific proteins so we can have full proteins there. The problem is, in the case of, for example, RGD, adhesion peptides, these need to be displayed at a fairly high concentration. So it was easier for us to display them, let's say, at a 1 millimolar concentration, if they are directly integrated on the backbone, rather than we have to catch them with our anchor strands. And these anchor strands are kind of precious to us. So that's why we kind of separate those tasks. But if there is a certain protein that can be active already at very low concentrations, we were thinking actually about certain growth factors, for example. They can be bound through aptamers also to the matrix itself. Yeah.
Okay, another question that was more about the forces. So in your last application for measuring the force from migrating cells, did you try different relaxation times to evaluate how cancer metastases can be regulated from mechanical tuning?
Yeah, it's a good idea, and we haven't done that yet. But it's I think a very, very good next step, because so this was done at a kind of, I think it was the 1-day relaxation time, and that was not varied. So all control that we have over the force probes, those are actually, we know that they open up at about, I believe the value is 3 piconewton. And this is something we can also change, but we didn't change the stress relaxation crosslinkers, which would make a lot of sense and probably would show different behavior of the cells as well. Yeah, very, very likely next step that we would like to take also.
Okay, thank you. The next set of two questions. First, said DyNAtrix is indeed a breakthrough system. So congratulations. Two questions. Is DyNAtrix compatible to perfusion? Have you tested that? Maybe we start with the first.
Yes, so that's actually something that that we believe that this should be an advantage, and it's in the grant application, and we want to do this. Yes, we haven't done it yet. In principle, perfusion through DyNAtrix should be more efficient because it's kind of very low binding affinities to biomolecules, as we see with LASSO, and has a very low solid content, actually. So in principle, we can put more inside, right? We could supplement it with additional, for example, laminin, but in its current state, diffusion is very fast through it. So, and we thought that this should also give certain advantages in terms of providing, nutrients or taking away metabolites from cells or spheroids or organoids.
Okay, and the second question was, given the synthetic nature of DyNAtrix, can it be coupled with extracellular matrix ligands like laminin to support immune cell migration studies, particularly in blood vessel organoid systems?
Yeah, I think we should get into that. It can. We believe it can. The simplest thing, what one can do, is supplementing things like laminin just to the matrix, and this can have a beneficial effect already. We know this from other studies and in preliminary studies that we have done. The ideal case, of course, would be, I think, as the question suggests, to have additional binding to those kind of additional supplements, right? To have laminin fibers that are bound to DyNAtrix and therefore don't kind of freely diffuse around and have other components that should work. I think there is a additional development that we would like to take where we have, let's say, click chemistry on the backbone that allows us to very easily add anything additionally to our synthetic backbone. Yeah. Good idea, very good idea.
Then another question, I don't know if you want to answer. Have you tried to use LASSO to capture specific small molecules?
Small molecules.
Yeah, small
We
molecules.
haven't, yeah. We haven't tried. It would be interesting if there is a kind of a killer application for that. Let's say, certain, I don't know, PFAS. Let's remove PFAS from something, right? That could be interesting. The question is, how can we kind of selectively capture those molecules? It could be, for example, we could have some ligands in there that binds toxic heavy metals or something like that. Or there are a lot of things that one could do if the application doesn't need a ton scale of the material, if it works on a small scale as well. That and it's very important. I think we can explore it. So if there are certain molecules you have in mind, so the person who asked the question, then I'd be happy to hear about that. Yeah.
Okay, so that will offer to the ad here that or add to the offer that you just made with a pilot users. Then another question: how do you produce an aptamer molecule for a specific protein?
So we don't produce aptamers ourselves. We rely on literature reported aptamers, and there are also companies that provide validated aptamers. And these companies can also just append the aptamer sequence with our own docking sequence for DyNAtrix. So we would tell that company, please give us the regular aptamer, but add the sequence that make it integrate itself into LASSO in this case. This is something that probably can be explored a lot in the future, because there is a lot of work one can do with aptamers, and one can also think about ways how we can not use aptamers but antibodies or nanobodies or any other target binding moiety to LASSO.
So this was, as far as I can see, the questions from the chat. If anyone else has questions, then you can also activate your microphone and speak up. I have an additional question about the fields of application. You mentioned like organoid systems like placenta, kidney, and brain, but also iPSC, which of course the stem cells are a big field nowadays in the let's say three-dimensional cell culture. Is there something like you are aiming at first, or where you see the applications more than in others, or?
So one thing we would be interested in at the moment is hepatocytes and cardiomyocytes, just because of the relevance in drug screening. So that's something we actually, we want to get into, sooner. And besides that, ideally, it would be great to have something that is really broadly applicable. So that's what we're trying to see, whether it's possible to have this broad applicability, because the big problem of people for switching away from Matrigel is the compatibility of all the other synthetic matrices. So everyone wants to go away from Matrigel, but in the end, they don't, because they have tried once with a synthetic matrix and it hasn't worked, and that was the end of the story. So in order to prevent that, I think it would be good to have a broad applicability. Yeah. So we are focused also on integrating more higher diversity of peptides. We have a couple of other peptides, adhesion peptides, that also bind to other integrins. And it's possible that a supplementation with laminin, for example, and in particular, is inevitable to have this broad compatibility. But in the end, since we are not biologists, we don't care specifically that much about any particular cell type or organoid type.
Okay. Nevertheless, maybe you can say, I mean, you already told also about some shortcomings of the system, but if you think about the research transfer, the translation, getting into more biological or physiological context, so what are the biggest challenges in getting this system running, like in a higher scale, or have you thought about that, or explored already some things?
Yeah, I think if the compatibility is solved, if the compatibility is really as broad as we want it to be, then I think applications in as a non-animal testing or newer approach methodology NAM area, that would be very exciting and could be very promising as one kind of large application domain. One kind of like the stepping stone application domain which we envisioned is any application area where actually current materials cannot do certain things that DyNAtrix can do, right? So for example, the mechanical adjustability, switchable stress relaxation, and things like this is interesting for people who work in mechanobiology and who recognize that the microenvironment not only is important for biochemical reasons, but also for mechanical reasons, right? And which want to answer research questions in this domain. For example, if you do research into cancer or conduct research with cancer organoids, it is known that the mechanics of tumor tissues is different than of healthy tissues. And in this way, you can answer a question, for example, in which direction does the causality go? And is there maybe a way how one can treat certain diseases by changing the mechanical properties of the tissue, right? Because cells respond to it. So these are applications that we're excited about.
Okay. Yeah, once again, thanks a lot. I don't see any further questions, but I think that was a lot that you told us and nicely illustrated. Thank you once again. If you're interested, then contact Dr. Krieg directly. You can also go through our office. And yeah, we'd love to hear many more stories about the platforms, and very good luck also with bringing your dynamic matrices exist project further. Thank you.
Awesome. Yes, thanks so much.
Bye-bye and see you next time on this channel. Thank you.
Bye.